Immunoglobulins are proteins produced by activated B cells in response to foreign antigens. These proteins, also called antibodies, play a role in the clearance of a pathogen and are present in the blood, bodily fluids, and throughout the tissues of the body. The five classes of mammalian immunoglobulins are IgA, IgD, IgE, IgM, and IgG. In mice, the IgG class is divided into four subclasses. The structure of an immunoglobulin consists of four polypeptide chains, two of which are heavy chains, and the other two which are light chains. Together, they form a tetrameric quaternary structure. The N-terminus portions of the immunoglobulin are the variable regions and are involved in antigen binding. Depending on the type of infection in the host, activated B cells will secrete one of the five types of antibodies into the body. The presence of an antibody type in the blood, such as IgG, would indicate a humoral immune response. In this experiment, we are analyzing the blood sera that was extracted from mice previously inoculated with Streptococcus pneumoniae. Using ELISA, we can quantify the unknown concentrations of IgG in the blood of the mice. ELISA stands for Enzyme-Linked Immunosorbent Assay and is a method of analysis which detects specific antigens with the use of antibodies. These antibodies capture the antigen, in this case IgG, to create a sandwich complex that is conjugated to the enzyme horseradish peroxidase, or HRP for short. The enzyme will produce a signal when exposed to the substrate TMB. The signals are measured as optical density units and are proportional to the quantity of IgG in the sample.
Our serum samples were diluted to two solutions of 1 to 1,000 dilution and 1 to 10,000 dilution. These samples were added to the respective wells of the pre-blocked strip and incubated for 60 minutes at room temperature and covered from light. After washing, 1X enzyme antibody conjugate was added to each well and incubated for 30 minutes under the same conditions as the previous incubation. After washing, TMB substrate solution was added to each well and incubated for 10 minutes under the same condition as before. The reaction was stopped with stop solution and the absorbance was measured for each well. And the absorbance was measured at 450 nanometers. This bar chart shows the concentration of IgG in infected mice and naive mice at different dilution factors. So if we first look at the dilution factor of 1,000, the infected mouse sample had a concentration of approximately 350 nanograms per milliliter, whereas the naive mouse sample had only around 200 nanograms per milliliter. And this pattern is similar to what we see with the dilution factor of 10,000. However, the concentrations are much lower, only maxing out at around 40 nanograms per milliliter for the infected mice.
Discussion. Looking at the IgG concentration between the infected and the naive mice from the results section concluded that the infected mice have a higher result of the IgG concentration than the naive mice. IgG performs well in terms of fixing complement and enhances phagocytosis by macrophages. There is immune response present and visible to the mice that are treated with S. pneumoniae virus. This shows mostly on the dilution factor of 1000. B cell antigen binding may have activated an IgG concentration in comparison to the naive mice. These B cells may have initiated antibody production to opsonize the S. pneumoniae, resulting in activation of complement and macrophage phagocytosis. However, in the dilution factor of 10,000, there's not much of a response, and the naive and the infected mice have an almost close result, knowing that there's no infection present in the naive mice. This could be due to some experimental errors such as calculating the dilution. Also, The naive mice could be having some type of pathogen that we did not know about. The high presence of the IgG can give a good idea of how pathogen was encountered, and this is the most common antibody isotype.